A very warm good morning to each one of you over here. So, today we are going to we are live with the second part of the chapter Principles of Inheritance and Variation for my dear sweet NEET 2022 batch. Super duper ready, everybody. So, let's get started with the inheritance of one gene. Monohybrid cross is our topic of the day. So today we are going to discuss about the monohybrid crosses, some of the questions related with today's class and the previous class on Mendel's hybridization experiment and the characters that he chosen for his experiment and based on the test cross, back cross, etc. Okay, so without wasting any time, let's get started with today's super amazing class. Those who are watching the session right now, don't forget to hit the like button, do share it with your friends, and subscribe the amazing platform for more updates on biology. And this is the most important thing which I have to tell you. BioPoint biggest surprise ever for NEET 2022-2023 batch. See you today sharp at 2:30 p.m. in the afternoon with this biggest surprise reveal about the mock test which is going to be conducted. Yes, so stay tuned for this amazing surprise. Everybody, do stay connected with our Telegram group and do stay attend this session. Register yourself for this biggest mock test which is conducted all over the India. Yes, everybody. So let's get started with today's session on genetics 1. Further details of this NEET mock test will be done at the 2:30 p.m. session. Yes, everybody, and we'll conduct a meeting on Telegram as well based upon this session. So let's get started with today's session on the Mendel's laws of inheritance, and let's get started with the very first term or the very first topic, that is the inheritance of one gene. Okay, so today we are going to discuss about the inheritance of one gene. Yes, everybody. Yes. So what do you mean by monohybrid cross? What do you mean by this monohybrid cross, everybody? Monohybrid cross means we are taking only one particular trait of character. Okay. We are taking only one particular trait of character for determining a cross. For example, we are taking a tall plant and we are crossing it with a dwarf plant. Or we are taking a plant with a violet flower and is crossing it with a white flower, or we are taking a yellow seed and is crossing it with a green seed. So that is referred to as a monohybrid cross. We are taking only one character at a point of time and we are doing the cross. A cross that is involving two plants differing in one character pair. Either it may be the difference in the length of the plant or it may be a difference in the color. Okay, difference in the color of the plant. Yeah, Debajit, every details of the test will be conducted or will be explained in detail. Each and every general instructions, key, everything would be made to clear for you sharp at 2:30 today. Every detail will be done and any doubts pertaining to the test after the session will be done as a meeting in the Telegram. Done? Yes. So, a cross involving two plants differing in one character pair is referred to as monohybrid cross. For example, we are taking a plant with tall and a dwarf trait and we are making to cross over it to study the inheritance of one gene. And what is the gene inheritance that we are studying over here? The inheritance of the height of the plant. Yes, here we are studying about the height of the plant. For example, we are taking a tall plant and is crossing it with a dwarf plant. Okay, tall plant, the gene is capital T, and the small plant, it is small t, or the dwarf plant, it is small t. Clear? So we are making a cross between a single trait, that is here we are taking or considering only the height of the plant, and hence we are coming to a conclusion about the inheritance of one gene only. Clear? Yes, everyone? Yes. 1 second. Yes. Suppose, listen very carefully. We are taking a homozygous tall. We have already studied what is homozygous, what is heterozygous in the last class. Okay, so homozygous capital T and homozygous dwarf, that is small t. So we are taking a plant or the parent with capital T trait and small t trait. Okay, heterozygous means capital T small t would stands for what condition? Heterozygous condition. Okay, capital T small t stands for what? Heterozygous condition. Here we are taking a homozygous parents. So what is a gamete obtained? So capital T from one parent and small t from the other parent. Therefore, we are getting the F1 generation as a heterozygous tall plant. Okay, heterozygous tall plant. Clear? You have to listen very carefully. If at least a dominant gene, if at least one dominant gene is present in a character, it will express the characters of this particular dominant gene itself. Okay? Yes. So since capital T is a dominant trait for tallness and small t is a recessive trait, since one capital gene is present over here, this particular organism will exhibit the characteristic of this dominant trait only. Okay, at least some of you may in some of your minds a doubt will arise like that. Why is small t not expressed? We already discussed that in the last class. Small t is actually referred to as the recessive trait. Okay, it is referred to as a recessive trait, whereas capital T or the gene which is expressed with a capital letter, easily identifiable, okay, it is referred to as a dominant trait. So in the first filial generation or the F1 generation, we are getting a heterozygous tall plant. So then we are doing what? Then we are doing the selfing of the F1 generation, that is capital T small t is crossed with capital T small t. Then you will get one capital T, two capital T small t and one small t. Clear everybody? Clear everyone? 1 second, let me take out a blank slide for you so that I can explain it to you very clearly. Yes? 1 second. Oh my god. Okay, guys, listen very carefully. I'm taking a homozygous tall parent and is crossing with a homozygous dwarf parent. Okay, so after that, crossing these two, I'll get the F1 generation as capital T small t. This is my F1 generation. Okay, then I'm crossing this, selfing this F1 generation, or I'm pollinating this F1 generation. That is, next cross I'm going to do is capital T small t into capital T small t cross. Yes, Calvin, we discussed about the cross. Yes, if you have any confusion, just ask me. Okay, so capital T small t, capital T small t. Here we are going to do the cross. Okay, here we are going to do the cross. Yes, everyone. So capital T stands for this gene or trait. Here it is capital T small t. Here it is capital T small t. Here it is small t. That is actually the cross. So let's discuss one by one. This stands for tall trait. This stands for tall trait. This is again tall. This is the only dwarf plant, or this is the only dwarf gene or trait which is present. Is it? Yes. So what is actually genotype and phenotype? We discussed yesterday, right? What is genotype and what is phenotype? We discussed in the last class. Genotype means it is a genetic constitution. Genotype is the genetic constitution and phenotype is the physical expression. So here when we are looking about the physical expression, you can see that 3 plants are tall, 3 plants are tall and 1 plant is the dwarf. Okay, 3 plants are tall and 1 plant is a dwarf. Devajit, are you attending the KVPY examination? Then this will be really useful. Okay, so 3 plants will be tall and 1 will be dwarf. So therefore the phenotypic ratio will be 3:1. So phenotypic ratio for a monohybrid cross will be 3:1. Okay, 3:1. Calvin, are you understanding, or should I repeat the points once again? Yes. Whereas the genotype you can call one capital T is there, two capital T small t will be there, one small t will be there. So the genotypic ratio for monohybrid ratio or genotypic ratio for a monohybrid cross is 1:2:1. Yes, very easy one. Okay. So in a monohybrid cross between two homozygous parent, only one particular offspring obtained as recessive, or one particular is obtained as dwarf. Clear? So that is what is discussed over here. So you have to read the NCRT very carefully about this Reginald C. Punnett, who was a British scientist. Okay, who was a British biologist. He was a scientist who proposed the Punnett square in order to explain about the probability of a cross. Okay, probability of a cross. Very important NCRT only. For NEET 2023 batch, from tomorrow we will be starting in today afternoon. We will be announcing the portions of the All India mock test. Okay, portions of the All India mock test. Today we at 2:30 p.m. we'll be announcing the date, time, and the portion, and the registration link for the thing also will be posted in the description and also in the comment section as well. So after the portion, we have 7 chapters in biology for our 7 chapters are there. So we will be starting the revision of these 7 chapters from tomorrow. Okay, from tomorrow for NEET 2023 batch, we'll be doing the one-shot revision along with the question discussion of those chapters. Clear? So we are going to rock the exam. We are going to compete with 2022 batch, 2023 batch are going to compete together. Clear, everybody? Yes. So inheritance of one gene. That is the F1 generation, that is capital T small t, when self-pollinated. Okay, when they are self-pollinated, they produce gametes in equal proportion. They produce the gametes in the equal proportion. Can you hear me, guys? Am I audible to everybody? Everyone have to write. Okay. Yes, I hope I'm audible to everybody. Okay. So during fertilization, pollen grains of T have capital T is 50% chance, and you also have the pollen grains of the genotype small t, that is recessive gene and dominant trait. Recessive gene and dominant trait is 50%. I think Calvin is not here. I'm not receiving any comments. Okay, 50 percentage equal proportion. You don't have to view over this binomial expression, all those things. You don't have to. You just have to. Whenever you in the question they have given you two parents like this, you have to understand what is the product in the F1 generation, what is the product in the F2 generation. You have to make the cross and you have to understand that. Okay, everyone? Yes. So this is what we have told. This is what we have told. Okay. So the parents we have taught, and this is also the parents. So you are getting 3 tall plants and 1 dwarf plant we are obtaining. Thereby we have come to the conclusion that the monohybrid phenotypic ratio, sometimes these questions may come as such as a direct question from the NCRT. 3:1, that is 3 tall is to 1 dwarf. Whereas monohybrid genotypic ratio is 1:2:1. You have to study which all are these. Clear, everybody? Clear, everyone? Is this clear? Perfecto. So let's move on. Yes. So this is what we have studied right now. that is a Mendel self-pollinated the F2 generation and found that there was the formation of dwarf plant as well. Even though Mendel took a cross between two tall plant, that is heterozygous tall plant, he found out that one plant which was obtained at the end of the or the product of the monohybrid cross was a dwarf plant. Okay, and Mental made similar observation for other traits as well. He done the experiment with flower color, he done the experiment with seed color, he done the experiment with seed shape, pod color, pod shape, flower position, and plant height as well. And he proposed the Mendel's first law of inheritance. He proposed the first law of inheritance that some factors were inherited from the parents to the offspring. He was not able to identify what was that factor, so therefore he termed that particular substances are transferred or inherited from the parents to the offspring by the term of factors. And what is this factors known by which name today? What is the factors known today? Yes, tell me, what is the present name of the term called factors? Yes, it is called the genes. Okay, it is called the genes. Okay, guys, everyone. Yes. Next, we are going to discuss about two important thing. Okay, two important thing, that is a back cross and the test cross. Okay, back cross and the test cross. Very good, Anandu, Debajit, Calvin. Very good. Yes, back cross and test cross we are going to discuss right now. Yes. So, guys, what do you mean by a back cross? What is a back cross? It is a cross between a hybrid variety and its any parent. Okay, guys, suppose in the last experiment we crossed between capital T and we crossed it with small t, right? And we obtained the F1 generation as capital T small t. Am I right or wrong? Yes. Am I right or wrong? Right, correct? Yes. So what do you mean by a back cross? Back cross means that I'm taking this particular offspring. I'm taking this offspring obtained as a result of the cross, and I can either cross with this capital T, or I'm crossing with this small t to obtain the F2 generation. Okay. And then I'm going to do the obtain the F2 generation. Clear? Yes. And what is the difference with the back cross with the test cross? Test cross means that we are crossing an organism with the dominant phenotype to the recessive individual. Okay. That is, in case of this particular test cross, we are taking this particular thing. Okay, this offspring is capital T small t, and we are crossing it with the recessive parent. If a cross like this appears in your option, then that stands for the test cross. This was a previous year NEET MCQ. Previous year NEET MCQ. Which of the following is test cross? Which of the following resembles test cross? Then you have to look for the parent, that is the F1 generation, is heterozygous. It is crossed with a homozygous recessive trait. Does anybody needs me to speak about the back cross and test cross once again? Or else we can move forward. Back cross means you are crossing the phenotype. Sorry, you are crossing the offspring either with the homozygous tall or homozygous dwarf parent. But test cross means the F1 generation have to be definitely crossed with the recessive parent only. Yes, Debojit, Calvin, Anantu, everybody have to tell me whether you have understood the topic or the concept of back cross and test cross very clearly. This is very important. Previous year question have came from this area. Just tell me fast, everybody. Yes, I'm not receiving any comments. What happened? Yes, what about Calvin? Yes. So the monohybrid test cross ratio is 1:1. Okay, monohybrid test cross ratio is 1:1. So whenever you see an option like this in the question, that is 1:1 stands for, then you have to definitely goes for the option monohybrid test cross ratio. Dihybrid test cross ratio is something different. Okay. Yes. So test cross is used to find out the unknown genotype of a character. If we doesn't know, if we have to find out the genotype of a character, then you are using the particular cross named as test cross. Important previous year NEET MCQ question. Name the type, or which of the following type of cross is used, which of the following type of cross is used to find out the genotype of an unknown offspring or unknown product? Then which is a cross? It is a test cross. How we are doing the test cross? The test cross is done between the F1 generation or the offspring obtained in the F1 generation along with the recessive parent that we have taken. Clear, everybody? So as we have already discussed, what is the Mendel proposed two important law? Mendel proposed two important law based upon this monohybrid cross. Okay, and one law based upon the dihybrid, and one based upon the dihybrid cross as well. Okay, so what is the first law? Law of dominance. Which was the first law of Mendel? First law of dominance, or law of dominance. He told that the characters are controlled by discrete units called the factors, and factors always occur in pair. If there is a gene for capital, then there would be a small t as well. One dominates and the other becomes recessive. This is referred to as the law of dominance. Clear? Sometimes simply they can ask you a question like law of dominance is based on Mendel's conclusion on. Options will be monohybrid cross, dihybrid cross, trihybrid cross, none of these. Which is the answer? Which is the answer? Just tell me. Law of dominance is based on which cross? Is it monohybrid, dihybrid, or trihybrid cross, or none of these? Which is the answer, guys? Very good. It is based upon the monohybrid cross. What about the second law? From the second law, there mostly we don't expect any question, but because this just our NCRT tells us only about the surface, whereas we have to study in detail, which we learn in the higher classes. What is this law stating? That is that during gamete formation, okay, during the formation of gamete, what happens? The factors are present in the parent segregate from each other. That is, if it is occurring as capital T, it will split up into offspring. Okay, it will segregate from each other. It will segregate from each other into the respective gametes. Okay, homozygous parents produces similar gametes, whereas heterozygous parent produces two different types of gametes. Suppose we are taking this as the parent, then we will getting similar gametes like this. If you are taking a gametic like this, then we'll get gametes like this. If you're taking heterozygous or dissimilar pair, we'll get dissimilar gametes only. I hope it's perfectly fine and clear for everyone. Yes, everybody, is it fine and clear for everyone? Just give me hearts on the screen, everyone. Come on. Yes, fill the chat box with the hearts. Come on, everyone. Yes, everybody. So let's get started with some of the question. We will be doing some question and we'll wind up today's session. Okay, we'll wind up today's session by summarizing everything that we have learned in the last class and today as well. So read the question very carefully, then comment out your answer. Mendel has chosen dash for his experiments. He selected dash pairs of true breeding pure lines. Answer on the chat box, everybody. What is Mendel chosen? Pisum sativum or the garden pea. Yes, Mendel selected garden pea for his experiment. He selected how many pairs of character? 7 pairs of character. Or he selected 14 true breeding variety. Since the term pair, they should listen very carefully. They have asked you how many pairs. Total he selected 14 and therefore we have to write 7 pairs. Clear? So the correct answer is option number 4. Clear? So Mendel has chosen garden pea for his experiment. He selected 7 pairs of true breeding pure lines. Question number 2. The color-based contrasting 7 pairs, the color-based contrasting trait in 7 contrasting pairs studied by Mendel in the garden pea plant where? We have already studied that Mendel studied 7 contrasting pairs of character. So how many of them were color-based? Violet and white. Violet and white, flower color. Yes, violet and white, flower color. Green and yellow, pod color. And finally you have the yellow and green, seed color. So number 1, 2, 3. So the correct answer is option number 3. Is it clear, everyone? Is it clear? Clear? Yes. Let's move on with the third question. What is the probability of the production of dwarf offspring? What is the probability of production of dwarf offsprings in a cross between two heterozygous tall plant? Two heterozygous tall plant. Can we start doing the cross, everybody? Two heterozygous tall plant, that is capital T, small t, is made to cross with capital T, small t. What is the probability of the dwarf plant obtained? Let's draw the probability square or the Punnett square or the checkerboard based upon this. I haven't find Calvin in today's class. In between, you'll come and comment something, but I'm not finding your answer continuously. Yes, you're asked to find out the percentage of the dwarf. You are asked to find out the percentage of the dwarf plant. That is capital T, small t, capital T, small t. Okay, let's do the cross. Capital T, capital T, capital T, small t, capital T, small t, small t, small t. So how many dwarf is there? Only one. So what is it? 1 by 4 into 100. 1 by 4 into 100 means 25 percentage. So the correct answer is option number 3. Very good. Yes. Okay. Let's move on with the fourth question. Which of the following indicates a test cross? Which of the following indicates a test cross? We have already told that the test cross is used to find the unknown genotype. Okay, we already studied that test cross is used to find the genotype of an unknown product. And we studied that the offspring obtained in the F1 generation is made to cross. With the recessive parent. So which is the option? This is the option. So option number 4 will be the correct, right? Right, everyone. So test cross stands So guys, thank you. I hope till the last question my audio was perfectly fine. I hope after we completed everything only I think it started breaking, right? Okay, guys. So that was the end of today's class, and tomorrow we'll discuss with the dihybrid crows. And don't forget to attend the session today at 2:30 p.m. It's a very important session. You all have to attend the mock test. Okay. Bye bye, guys. See you.